This paper is incredible. The title is Cortical-blood vessel assembloids exhibit Alzheimer's disease phenotypes by activating glia after SARS-CoV-2 infection. Now, they bring up a really important point that brain organoids are insufficient in some ways because they don't have immune cells and they're absent vasculature. To compensate for this, they came up with a novel method where they bind a cortical blood vessel assembloid with the cortical organoid with the blood vessel. So basically, they fused a blood-brain barrier type thing around these nerve cells, and these are all human-based. Now, let's take a moment to appreciate how freaking cool that is. What they found is that beta-amyloid plaques are affected by the inflammatory response of a SARS-CoV-2 infection, where this neuroinflammation caused by the COVID virus is facilitating an Alzheimer's disease pathology. So some quick background, neuroinflammation has been revealed as a key role in the pathogenesis of Alzheimer's disease, with the hallmarks being these beta-amyloid plaques and hyperphosphorylated Tau Tangles, with the neuroinflammation being sort of defined by glia with overproduction of pro-inflammatory cytokines, which proceed to cause this alpha-betaloid deposition and causes neuronal death subsequently. So with this fused brain model, they were able to create blood-brain barrier-like characteristics with tight junctions and adherence. And then they incubated these fused brains with COVID. And then they looked at the brains about 4 days after infection, where they saw infected cells in these blood vessel regions and showed that COVID induced neuronal cell death, partially by creating a locally hypoxic environment, so restricting that blood flow, and that even neurons in cells that were unaffected created an indirect effect where brain death occurred around that infected area due to locally induced hypoxia, which looks like this here, where you have these diffuse areas of cell death. They saw an increase in these tau level expression and an increase in amyloid beta accumulations in these COVID patients. And they tested for amyloid beta 42 and 40, which promote fibril formation, showing that these were significantly increased after infection. So here's the alpha-betaloid 42 comparison between controls and then those infected with COVID, as well as the 40 comparison. So you can see that the ratio is significantly increasing. And then again, there's an increase in these phosphorylated tau markers from the control to the COVID. And then this is what it looks like. So you have very low levels of these in the control, and then very high levels here in the SARS-CoV-2 patient, which provides another piece of the puzzle, showing us that this neuroinflammation caused by COVID can result in Alzheimer's-like pathology.